Let's talk about hem-onc anatomy. Hematopoiesis. Well, in hematopoiesis, we have the multipotent stem cells, and you have to know that the multipotent stem cell has a CD marker, which is CD34. They are the CD34 positive cells. In multipotent stem cells, they are converted into, or they can, they're going to convert into myeloid stem cells and lymphoid. In the myeloid stem cells, they are going to produce the myeloid cells, which are erythroblast, the megakaryoblast, all the myeloblasts and monoblasts. Erythroblast is called erythropoiesis. Let's talk about this thing here. Erythropoiesis, what is that? In the bone marrow, erythroblast is formed and it's going to convert it into reticulocyte. And that reticulocyte goes and gets converted into erythrocyte, which goes into the blood. But if the erythroblasts are making so many RBCs, they sometimes even send the reticulocytes outside in the blood, and we can see the reticulocyte count. And I think it's going to come up later. But erythroblast, reticulocyte, and erythrocyte, and all, that's all. For thrombopoiesis, we have the megakaryoblast. Megakaryoblast becomes megakaryocyte. Megakaryocyte is still in bone marrow, but as soon as they convert into the blood, or they come to blood, it's called platelets now. They're the platelets. So thrombopoiesis, platelet formation. Granulocytosis, cytopoiesis. Granulocytopoiesis. We have the formation of myeloblast. What are the myeloblasts? They're the, let's talk about from here, band formation. Bands are the type of neutrophils. So you will see in the other sections that we will be talking about bands, neutrophilic bands. So those are the bands right here, which are present in the bone marrow and normally, and in the blood they will be coming and forming formation of neutrophil, conversion into neutrophils. You have the basophils. Looking at the picture, neutrophil has plenty of lobes. Okay, but the lobes are important in megaloblastic anemias greater than 5. Is the one thing that we can look at for the in the megaloblastic anemia. But coming back here, we have eosinophils, pink cells, basophils, blue cells. Okay, maybe this looks like a headphone. I don't know. Headphone shaped. Okay, but basophils, and also one more thing, when they come into the tissues, now they're called the mast cells. Mast cells. Okay, now let's talk about the monocytopoiesis. Monoblast will when they come to blood, called monocyte, and when they go to tissues, called macrophage. Macrophage, dendritic cells, and you can name certainly all of those, but dendritic cells, cover cells, all are macrophages, but different tissues. Okay, the other thing now, let's talk about the lymphoid cells. The lymphoid cells, lymphopoiesis, the lymphoblasts are going to convert into the B cell, the T cells, and natural killer cells. B cells, they will form plasma cells. T cells they will form T helper cell and T cytotoxic. So CD4 cells and CD8 cells, right? And these plasma cells here, they are gonna form the antibodies, AB's antibodies. And what else? In CD4, we have two cell types in there. We have the T helper 1 cell and T helper 2 cell. And these are all the multipotent stem cell divisions. One more thing, let's talk about WBCs. WBC being WBC or leukocytes. In leukocytes, we have two sorts of classification. We have one. We have one which is called granulocytes, and the other one, the whole this thing area. These are agranulocytes. Okay, and remember the macrophages are the agranulocytes. So in the granulocytes, we have the eosinophils, the basophils, the neutrophils, the mast cells. And in the agranulocytes, we have the macrophages, the B, T, and K cells. Okay. Now let's talk about neutrophils. Neutrophils, they're going to come in the acute inflammation response cells, phagocytic, multi-lobuled nucleus, which is looking a multi-lobuled nucleus. Specific granules contain leukocyte alkaline phosphatase LAP. Okay, and collagenases, lysozymes, and lactoferrins. Azophilic granules, lysozymes contain proteinases, acid phosphatase, myeloperoxidase, beta-glucuronidase. Inflammatory states like bacterial infection causes neutrophilia and change in neutrophil morphology, such as left shift and toxic granulation. Look at the toxic granulation. Left shift toxic granulation, dark blue, is going to be dark blue coarse granules, and Dolly bodies, which are the light blue peripheral inclusions, arrows in A. So peripheral inclusions, light blue peripheral inclusions, which are the Dolly bodies, and cytoplasmic vacuoles. Neutrophil chemotactic agents are very important: C5a, IL-8, LTB4, and 5-HTE, which are the leukotriene precursors. Also, kallikrein, platelet activating factor, PFs, and from bacterial, you have the N-formylmethionine, which are the bacterial proteins. So these are all the things. They are the chemotactic agents for neutrophils. Hypersegmented neutrophils, 6+ lobes, are seen with either vitamin B12 or folate deficiency. Now we did talk about left shift, so let's talk about what is left shift. Left shift means you have an increase in neutrophil precursors. Example, the bands. Remember we talked about bands as a precursor. Look in here. Band is a precursor that is coming from the bone marrow. It's still a precursor to neutrophil. So what do we have in the left shift? We have a lot of bands and a lot of metamyelocytes. In peripheral blood, they just come into the blood because the body's making so many of them. It's like, throw them out, man. It's really hard to mature them here. Okay, so they throw it out in the periphery. That's called left shift. Why do we have the left shift? Well, it reflects the states of increased myeloid proliferation. Example, either normally in inflammation but pathologically in CML. Pathologically CML, normally inflammation. Inflammation causes a lot of neutrophils to be produced, and that's called left shift, in which you will see even the bands in the bloodstream. Now let's talk about the Leukoerythroblastic reaction. That's the left shift accompanied by immature RBCs. So leuko means white blood cells, erythro means RBCs. Blastic reaction, that means so much are being produced So that means you have the neutrophil and RBCs are being produced so much that the immature cells are in the bloodstream. So just bone marrow infiltration, like in myelofibrosis, when you raid the house, this was the house for them. This was the house for neutrophil and the RBC. When you raided that house, now you can't increase the walls of the house, right? So there's only that specific space. In that space, you have the myelofibrosis and metastatic disease to be present. They're kicking out their own residents. They're like, kick you, man. Neutrophil, get out of here. Get off RBCs. But these are not fully matured neutrophils and not fully matured RBCs. Okay, so immature neutrophils, immature RBCs, known as leukoerythroblastic reaction. Okay. So what did we learn here? We did learn that in the inflammation, if we have an inflammatory state, we're gonna have a left shift. We're gonna have the toxic granulation, that the change and the color dark blue and coarse granules will be present. We saw that we also gonna have the Dolly bodies. We're gonna make some cytoplasmic vacuoles, and this is all in the inflammatory states. This is all gonna happen. Okay, for in the neutrophils. Okay, let's move on. And the thing is, let me also one thing here. How does it kill something? Specific granules, they release they fuse with the phagosome. So specific granules, first of all, they release some extracellular things that was going to kill something, whatever they're killing bacteria, and they will fuse with phagosome, and this, as soon as they fuse with the phagosome, you kill the pathogen within the phagosome. Okay. So again, simple thing. Specific granule has all of these things. They release all of this in extracellular matrix, and they themselves then fuse with phagosome. And same thing with the A's or phytic granules. They also fuse with the phagosomes and destroy whatever they have captured. All right, moving on. What do we have? We have the erythrocytes. Erythrocytes are simple. They carry O2 to tissues and carbon dioxide to the lungs. Yeah, simple. Anucleate, very important. Anucleate and lack organelles. Biconcave. Look in the picture A. With large surface area to volume ratio for rapid gas exchange. Lifespan is 120 days in healthy adults, 60 to 90 days in neonates. Source of energy is glucose, 90% used in glycolysis, 10% used in HMP shunt. So they can only take glucose. 90% used in glycol. That glucose is used in glycolysis, and 10% goes to HMP shunt. Membrane contains chloride bicarb antiporter, which allows the RBCs to export the bicarb and transport carbon dioxide from the periphery to the lungs for elimination. Erythro is red, cyte is cell. Erythrocytosis equals polycythemia, basically increase in RBCs, which increases the hematocrit. Anisocytosis, varying cells. reticulocytosis, varying shapes. Reticulocytes are the immature RBCs that reflect the erythroid proliferation. Bluish color, polychromasia, or on the right Giemsa stain of reticulocytes represent residual ribosomal RNA. Okay. And when do we see these residual ribosomal RNAs? If we have a non-functional spleen or in splenectomy patients. Okay, coming to the next thing, we have thrombocytes. Thrombocytes are the platelets involved in the primary hemostasis. Anucleate, again anucleate, small cytoplasmic fragments derived from megakaryocytes. Lifespan of only 8 to 10 days. Platelet 8. When activated by endothelial injury, activate with other platelet interact with the fibrinogen to form platelet plug, and this will be coming up in the pathology things. Contain dense granules. What does it contain? Dense granules, which contains calcium, ADP, serotonin, histamine. Cash is the mnemonic. But remember, it has dense granules and alpha granules. Dense granules contains the calcium, ADP, serotonin, and histamine. Alpha granules contains the von Willebrand factor, the fibrinogen, the fibronectin, the platelet factor 4. Approximately 1/3 of platelet pool is stored in the spleen. Okay. Thrombocytopenia or decreased platelet function results in petechiae. So either you have a decrease in the platelets or decrease in the function of the platelets, you're going to have a petechiae. Okay. Von Willebrand factor receptor is the GP1B. Fibrinogen receptor is the GP2B3A. Thrombopoietin stimulates megakaryocyte proliferation. Alpha granules contain von Willebrand factor, fibrinogen, fibronectin, platelet factor 4. So all the F's. Alpha, von Willebrand factor, fibrinogen, fibronectin, platelet factor 4. Okay, moving on with the monocytes. That is found in the blood and differentiates into macrophages in tissues. Large kidney-shaped nucleus, extensive frost glass cytoplasm. Looking in the picture, kidney-shaped nucleus with the frost glass cytoplasm. Kidney-shaped nucleus is the macrophage. Mono equals 1 nucleus, site equals cell. Macrophage now, it's a type of antigen APC, antigen presenting cell, phagocytose the bacteria and the cellular debris and the senescent RBCs. So who kills the RBCs? Macrophages. Long life in tissues. They're differentiated from circulating blood monocytes A. Look at the picture A. So they're differentiated from circulating blood monocytes. So looking at a monocyte sort of gives off the formation of macrophage, as we know that, and that's going to be looking like a kidney-shaped cell. It is activated by, very important, IFN interferon gamma. Okay, can function, this is important, can function as an APC via MHC2. APC as an MHC2. Also engage in antibody-dependent cellular cytotoxicity. So they can present the cells. They can also engage in antibody-dependent cellular cytotoxicity. Important cellular component of formation of granuloma, which is formed in the tuberculosis, sarcoidosis, okay, and fungal infections, and cat scratch disease, Bartlett-Hensler, where they may fuse to form giant cells. So what they're saying is, these macrophages, they fuse together, and now they're called giant cells. Okay. macro large phage eater. Macrophage names vary by specific tissues. Example: Kupffer cells in the liver, histiocytes in the CTs, osteoclast in the bone, microglial cells in the brain. So what are the names? The Kupffer cells, the histiocytes, microglial cells, and the osteoclasts. Okay. And then you have this lipid A from bacterial LPS bind CD14 on macrophage to initiate the septic shock. Okay, so you kind of get that from the lipid A coming from the LPS, from the bacterial LPS. It can initiate a septic shock by binding it to the CD14 off macrophage. So what was the function of this thing? Once it gets activated by interferon gamma, it is first it's doing antigen presentation, APC. Second thing, it was doing phagocytosis. And third thing it was doing is the cytokine production. Okay, and they are also doing the granuloma formation. That's all for the macrophages. Now let's talk about the dendritic cells. Dendritic cells are highly phagocytic APC cells. They function as a link between innate and adaptive immune system. Example, via T cell stimulation expresses the MHC class 2, again same thing, APC MHC2, and FC receptor on the surface. They can present to the exogenous antigen on an MHC class 1. Okay. So let's just a little bit talk about what do they mean by all that. So what are these dendritic cells? So these dendritic cells, they're constantly sampling their environment by endocytosis. What do I mean by endocytosis? They take up something and they process it down. So they take from outside, they endocytose that. Okay. And they will get stimulated upon entering the foreign antigen. So as soon as they say like, okay, whatever we endocytose, that's the foreign antigen, they get stimulated. When these dendritic cells get stimulated, because they were just sampling, right? Normally sampling their environment as they're just walking. So as soon as they get activated, they then migrate to the draining lymph nodes. Okay, and then it goes to the spleen. And from there, they display antigen, the same antigen they have the endocytosed, they bring it up, and they present it on their MHC class 2, where the T cells get stimulated. T and B cells, both of them get stimulated by that. Okay. And that's the same thing with all the APCs. But the difference is, they're just continuously walking and doing sampling. Okay. If these dendritic cells are in the skin and mucosa, we call them Langerhans cells. Skin and mucous membrane called Langerhans cells. Okay. Not the Langerhans giant cell, that is something else. This is Langerhans cells, I think, which is also going to come. And characteristically, you will see, very importantly, they will say, okay, racket-shaped cells. Racket-shaped cells. And it's going to be shaped like this. Okay, so these are the Langerhans cells. Now let's talk about the eosinophils. Eosinophils, we have defend against helminth infection, major basic protein, major basic proteins, bilobe nucleus, look at the nucleus, bilobe nucleus, packed with large eosinophil granules of uniform size and highly phagocytic. Okay.